And I'm on a grant with Mitch Gutman doing tool development, where we tried developing modified guides for live cell imaging with CRISPR, which lots of labs are doing. But I wanted to just give sort of an overview, since I haven't given an update yet, on what our progress was last year, and some new sort of questions that kind of emerged as we were comparing live cell imaging to HiC data. So on the first slide here, we're looking at HiC data of the fire locus across several human and mouse cell lines. We also have looked at this across species, and it's pretty well conserved, that there's a long non-coding RNA called fire, functional energetic repeating RNA element, that's located on the X chromosome right in the middle of two TAD boundaries indicated by the dashed lines in the middle. And below is a zoom in of CTCF binding. And so if you look at fire, the fire locus in its entirety, there's a wide, several binding events seeming to go on for CTCF in this locus. So this inspired, on the third slide, a very talented postdoc, Rinn, to come into the lab, and he sort of looked at fire genome-wide, is what's plotted here. And you can see in the first plot that for as far as density goes, fire is the red dot, and it's at the top of these distributions in the genome, same in mouse. And then also at TAD boundary strength, you can see fire is at the top of the list across many cell lines. So this guy looked like a great candidate for a non-coding RNA element that sits at the boundary of two TADs and is highly enriched for CTCF and resides at a very strong and conserved TAD boundary. So the logical thing Rinn wanted to do is use a genetic approach where we delete the swath of CTCF sites, shown here on the slide 4, by deleting the entire fire locus, including its promoter, which people are starting to realize are more and more like enhancers. So we removed all cis sort of regulatory functionality in this locus. We also made a gene where we put the fire cDNA, the spliced version of fire, on an autosome that's inducible, so we can see if we can rescue any defects in the knockout, and then combine these to have the knockout background with the inducible transgene in it. And then in slide 5, we're looking at what happened in the complete ablation of the fire locus, again, including its promoter. We can see that when on the left side, that when we knock out fire expression's gone. And also below that is a plot of HiC contacts being lost in the knockout, as would be expected. What was a bit unexpected is this middle plot of HiC data showing fire in the middle with the dashed lines. And you can see quite, almost perfect preservation of the TAD boundary that fire sits in, but also the surrounding area on the X chromosome. And we also did this allele specifically so we could rule out something between the X and the active and inactive X, and both in male and female cells. So pretty much on the right is the quantification of that somewhat intuitive visual result, that the insulation score doesn't change at all, nor does the TAD boundary strength where fire was removed. So despite removing roughly 15 CTCF binding sites, we didn't see any change in this specific region. And that's where we wanted to ask if maybe X chromosome inactivation was a defect that we wouldn't see by HiC. And in order to do that, we teamed up with Ginny Lee's lab. They did the exist and fire co-staining. So fire's in red in these images, and the exist RNA is in green. This is a heterozygote knockout, so we can ask, is the intact allele associated with exist more or less often than wild type? And basically we see no change in skewing. So the deletion fire did not change the TAD boundary locally, nor did it change the ability to randomly select inactive chromosomes for inactivation. So if we looked more globally, now here on slide 7, what you see is just wild type comparison knockouts of this so-called megadomain on the X chromosome depicted below in those bar diagrams, where fire sort of acts with several other elements to localize to a site called DXE4 macrosatellite region, and X75 ice and fire all sort of converge on this central point. This has been seen by many groups. It's a very fascinating, big, long-range genome architecture, if you will. And so what we can see in the knockout on the left side here is, if you look at the middle panel, that fire's gone, those interactions are gone, but we see a slight increase with interactions with X75 and DXE4 in the absence of fire. So there does seem to be some movement of the, where you can see this here is becoming more dense. And so we do see that when fire is removed, this somehow increases the occupancy of X75 at DXE4. And we looked at this on slide 8 by CRISPR Live Cell imaging. This is just basic MS2, PP7 modifications of guides, followed by conjugate fluorescent proteins. And so what we can see here is that in the cells, we do see fire in DXE4 and X75 co-localized, and we see somewhat of an increase of that in the knockout. It's quantified by this below on slide 9 is the quantification of this, where we can see that in the knockout, we see a significant decrease of fire X74 or DXE4 interactions, as we'd probably expect, and an increase of fire or X75 and DXE4 in the knockout. And that's depicted on this cartoon here, where we see X75 in the absence of fire likes to sample back into the DXE4 region more strongly. It should be noted that these interactions are not seen in all cells, and it's roughly anywhere from 15 to 30% of cells show this domain. Also to note, though, that only 50% of cells actually exist expression 2 during X inactivation. So we wanted to then see if there's any gene expression changes. There is this hierarchical modulation of X, small effect, but significant, of X75 moving differently. And we wondered if that affected local gene expression. And so slide 10 is showing you a plot of a wild type versus knockout fold change in expression. Fire is gone, but none of the genes are indicated by these gray and black dots in any of the tissues we looked at were affected. In fact, we don't see anything happening on the X chromosome as far as gene expression changes. That's further sort of reinforced on slide 11 here, where we can see that fire is gone in all of our knockouts. We look at all these tissues, and we don't see any of the local genes in a 2 megabase window changing. The same is true for the entire X. So summary 1 is a genetic approach at testing a very strong, conserved, high CTCF occupancy TAD boundary. Full gene ablation of that did not affect the TAD boundaries in this case. It did change a quaternary interaction, if you will, of this fire DXE4 switching to more DXE4 and X75 interacting, yet does not result in any changes in gene expression, nor does it change X chromosome inactivation skewing. So this, in parallel, sort of switching gears a little bit to the live cell imaging aspect, we've been interested because of our investigations of fire and this bizarre observation that fire seems to sit next to other chromosomes in the nucleus. That's sort of indicated here that we've just been discussing intra-chromosomal interactions mediated by fire. And now we wanted to look across chromosomes that we had previously mapped by pulling down the RNA and asking where it is in the genome, and also by live cell imaging, seeing co-localization of the X chromosome and autosomes. So it was just a quick overview of, there's many different flavors of this now. We basically just use it as a tool. It's CRISPR, we call CRISPR Live Cell imaging. There's other names. We're not very particular about the naming, but we can call it CLING for simplicity. And really the principle behind here is you take a guide RNA and modify the end of it to have an RNA epitope that's recognized by a cognate protein that's fused to a fluorescent protein, in this case indicating M cherry and M venus in red and yellow. So this is nice because you can program the guide to have a specific color. So one site in the genome has a specific color, and you can do multiple colors, only limited by the number of RNA, protein, and epitope interactions you can make. And we're working on some new technologies to expand that arsenal as well for our next phase of the grant. In slide 15, we further tuned this, tested, it's probably an obvious proof of principle, but it worked really well. We were very happy with it, where we tried to design guide sequences where the PAM would be disrupted by an allele-specific SNP. So we used an easy or more dramatic model for this, where we made a hybrid mouse between two different species that have a high density of SNPs, so we can easily distinguish between the maternal and paternal alleles. And this is shown here on the bottom, that we can see the maternal and paternal allele of YPL4 and individually in different colors. This was kind of fun, a fun proof of principle, where we, in this paper, looked at how these chromosomes move around in the nucleus relative to each other as sister chromosomes, as well as across other chromosomes of different properties like gene density, size, and other things. And what we found is that they're moving, but they're well oriented around nucleoli. Was an insight there, but this by being able to determine which allele is which was actually a really important aspect for our goal of understanding inter-chromosomal interactions, shown on slide 16, where we needed to know which allele was which, because if they are co-localized, the question is this a way of imprinting, where the maternal chromosome somehow knows to touch the maternal autosome, the maternal X knows to touch the maternal autosome, or is it mom, dad, or is it random? And unfortunately, what we found, we thought the hypothesis would be really interesting if somehow there was some allelic transvection between these guys, but we did not see that. Depending on how you interpret this, in my opinion, it's basically random, whether it's maternal or paternal pairings. And we've really only observed these things phenomenologically. This is shown on slide 17 here, where you can see the X chromosome labeled in red and this chromosome 11 gene YPL4 in yellow. And you can see that they're in close proximity. And we wanted to use our fire knockout model to see if any of these intrachromosomal interactions are actually changing in gene expression. We know their localization's disrupted, the knockout of fire. But what's interesting is we have 5 sites that we've sort of validated. YPL4 has always been one of the best ones. And what's interesting in the knockout of fire, in the bottom right corner here, you're looking at that sort of same cis window view in a fire knockout of full change of wild type over knockout. And you can see YPL4 is quite far down, but interestingly, so are all the neighboring genes. And this is actually the region where we see the RNA bound. From a previous study collaborating with Mitch Gutman and Jesse Engreitz, where we looked at where fire is bound on DNA outside of the X chromosome. And this happens to be a site that came down in RAP DNA methodology to find the cognate binding sites of fire. So that in one of the 5 cases, so who knows if that's just random, but it is quite striking that in the fire deletion, this autosome is completely affected and many genes in cis. So slide 18 is setting back up this thing that, okay, so we see these interchromosomal interactions in live cells in roughly about 50% of the cells. And when we have a test case now where we knock out fire, one of them is disrupted. And so indicating that they're possibly functional even. But we wanted to test why we hadn't seen this in HiC data. We'd been really trying to find some reads that had hybrid reads between chromosome X and 11 in this region and even broader regions, and couldn't really find it. So this sort of led to a more technical comparison where we took very deep HiC data sets and looked for intra and inter-chromosomal interactions. I apologize for duplicating the titles there, but on slide 19, the left side should say intra-chromosomal interactions, this mega loop we had talked about. These interactions are very readily detectable in HiC data. Even very long-range ones are very easily detectable as these red dots that appear, and in regions that we have not seen before to have interchromosomal interactions. We don't see any of these reads, which is consistent with many studies that have been published saying they don't see any interchromosomal interactions in HiC. Same here, where we can see clear co-localization in over 60 or 58% of the cells between fire and another site we've been looking at, ATF4. Again, this is not detectable over random things that don't co-localize in HiC data. So that made us question, like, why are these imaging and HiC data not sort of agreeing? So we took a bunch of sort of, I would call it, basic biophysical property measurements, where we sort of measured how fast these things are tumbling, moving apart, breathing, and they seem to be pretty stable, stay in proximity. However, they do appear to be at a further distance that we can measure with this Kling approach. And so one rationale we think why these things are not as readily detectable without capture or other secondary approaches is that the distance of intrachromosomal interactions is quite smaller as measured in microscopy than the interchromosomal interactions, not to mention all the chromatic proteins and other things that might prevent restriction digesting at these longer distances. And so that was our best conclusion for why we weren't seeing these things that we could see by imaging. So in summary, two inter and interchromosomal interactions are occurring about 50% of cells. We see random interactions of chromosomes in about 25% of cells. Either way, these are not observed in HiC. And the YPL4 fire interaction does appear to be disrupted by a fire knockout, but whether that is actually due to these interchromosomal interactions or some other mechanism, we don't yet know. But these are the people who will be certainly figuring this out. The last slide here is Philip Moss, who did a lot of the development of CRISPR Live Cell imaging, really just to test this hypothesis about potential maternal-maternal or paternal-paternal imprinting across chromosomes. And Rasim Barutu, who's on the phone, I believe, is now a postdoc with Ben Blencow, but did all of the fire knockout HiC data. And this guy is amazing. You can ask any question and he can give you an answer in about 10 minutes with HiC data. It's really been fun. And Chiara Gerninger is always our lab manager, involved in all of these projects. And Jordan Lewandowski is studying the mouse model of fire in the 3 different genetic configurations I mentioned, and has some very interesting findings of how the fire RNA is acting as a transacting RNA molecule in blood development. So with that, I guess I'm right on time at 9:20. I'll take questions.
That was right on time. So we have 10 minutes for discussion. Can I just jump in very quickly and ask, yep. Have you seen these trans interactions by Sprite as opposed to HiC?
Good question. So Sprite's amazing, and I think it's one example of a logic that I think a lot of us are starting to use, where you add a barcode, shuffle, add a barcode, shuffle, and the more representative barcodes you see for an interaction, the more real it seems. So Sprite is amazing because it finally finds the nucleolus. The 4 acentric chromosomes containing the ribosomal DNA repeats are known to interact in proximity near the nucleolus, and Sprite was able to detect that. So we really do want to apply that. I tend to let the postdoc go off and take the mission on this, and Philip Moss has started his own lab, and he's already up and running, and I'm sure is going to be collaborating with Mitch on these questions.